Okay, thank you, and thanks to the organizers for the invitation to speak here. I'm going to talk about how dormancy may induce speciation in a population model with a changing environment. This is joint work with Fernando Cordero and Adrian Gonzalez Casanova. So it's well known that in many populations, individuals will periodically enter a dormant state. Mammals that hibernate during the winter are one familiar example of dormancy. Many plants produce seeds which stay in a dormant state for a while before they eventually germinate. And it's also known that many bacteria are able to enter a dormant state. The primary advantage to dormancy is that entering a dormant state can allow individuals to survive periods of unfavorable environmental conditions. Now, it has also been suggested that dormancy could contribute to speciation. This recent paper of Blath, Paul, Tobias, and Wilke-Berenger found, at least in a certain population model, that dormancy could increase evolutionary branching. On a more applied side, in this 2011 paper, the authors found that dormancy may be contributing to a high rate of speciation among South American tomatoes. So our goal is to investigate how the fluctuations in the environment caused by seasonal changes may allow dormancy to induce speciation. And we're going to consider a population model that is similar to the seed bank model proposed by Blath, Gonzalez, Casanova, Kurt, and Wilke-Berenger in this 2016 paper. But we're going to add seasonal changes to the model. We're also going to allow individuals to acquire two different types of mutations. Some mutations are beneficial during the summer, but disadvantageous during the winter, and other mutations are beneficial during the winter, but harmful during the summer. And what we will see are two waves of adaptation. Some individuals will acquire many mutations that are beneficial during the summer, and others will acquire many mutations that are beneficial during the winter. The individuals that acquire many mutations that benefit them during the summer will still have a chance to survive the winter by entering a dormant state, and likewise for the individuals that are fitter during the winter. And so we see essentially what we end up with are two distinct subpopulations which are getting further and further apart genetically over time, and we're proposing that this could be a mechanism that contributes to speciation. So let's now formulate the model precisely. We have discrete generations and a fixed population size. So in each generation, there are capital N active individuals and K sub n dormant individuals. We'll sometimes refer to the dormant individuals as the seed bank. The environment alternates between the summer and the winter. Each year lasts for U sub n generations and consists of a summer lasting for V sub n generations, followed by a winter lasting for U sub n minus V sub n generations. So we have many generations in every year. Now, to incorporate selection, we'll say that each individual has a type, and the type will be an integer. An individual having type K has fitness 1 plus Sn to the power K during the summer, and 1 plus Sn to the power minus K during the winter. So positive types are beneficial during the summer, negative types are beneficial during the winter. Now, to go from one generation to the next, first we have some mixing between the active and the dormant populations. We randomly choose C sub n dormant individuals to become active, and we randomly choose C sub n active individuals to become dormant in the next generation. The other K sub n minus C sub n dormant individuals simply remain dormant in the next generation, and because they're in a dormant state, they do not reproduce. But the other n minus C sub n active individuals are new individuals born in this generation, and they choose their parent at random from the active individuals in the previous generation with probability proportional to fitness, so that fitter individuals are more likely to have offspring. And then finally we have mutations. With probability 1 minus 2 times mu sub n, the offspring will have the same type as the parent. but with probability mu sub n, the individual will acquire a positive mutation that's beneficial during the summer, and the type will be 1 greater than the type of the parent. And likewise, with probability mu sub n, the individual will acquire a mutation that is beneficial during the winter, and its type will be 1 less than the type of the parent. Now, we're going to need several assumptions on the parameters. First, we're going to assume that Sn converges to some positive number s as n goes to infinity. So remember that a beneficial mutation increases an individual's fitness by a factor of 1 plus Sn. So this assumption means that we're working with strong selection. Also, we assume that Vn over Un converges to some number beta between 0 and 1 as n goes to infinity. So remember, Vn is the length of the summer, and Un is the length of a year. So this is saying that in the long run, it is summer a fraction beta of the time, and winter a fraction 1 minus beta of the time, and in particular, the two seasons have comparable lengths. We're also going to assume that Un is much less than the log of n. By this symbol here, I mean that Un over the log of n is going to 0 as n goes to infinity, so in particular, the seasons cannot be too short. We also assume that Cn is much smaller than n. So Cn is the number of active individuals that enter the dormant population in each generation. So the assumption here is that it's only a relatively small fraction of the active population that is turning over in each generation. And finally, we assume that the mutation rate is not too slow. So in particular, there's some positive number b such that mu sub n is much larger than n to the minus b. That's actually a very weak assumption. Even a mutation rate of n to the minus 20 wouldn't violate that assumption. So to start to analyze this population model, let's think about what happens to the population after the first beneficial mutation. So initially, all of the individuals have type 0, and then let's suppose that before the end of the first summer, one individual mutates to type 1. Well, in the next generation, most of the active individuals are going to choose their parent at random from the active individuals in the previous generation with probability proportional to fitness. Now, the type 1 individual has fitness 1 plus Sn, and the other n minus 1 individuals have fitness 1. So the probability that type 1 is chosen as the parent is given by this fraction here, which is approximately 1 plus Sn over n. Now, there are n minus Cn active individuals that are born in the next generation, and because Cn is much smaller than n, that number is approximately equal to n, and so the number of type 1 individuals in the next generation will have approximately a Poisson distribution with parameter 1 plus Sn. And this process continues in subsequent generations, so for a period of time, the number of type 1 individuals will be well approximated by a Galton-Watson branching process, whose offspring distribution is Poisson with parameter 1 plus Sn. Now, this branching process may survive forever, or it may go extinct. If it survives forever, then that means that most of the active population is going to be type 1 after a short period of time. And by a calculation that goes back to this early work of Kimura and Ota, the time that that takes is of the order log n. On the other hand, if the branching process goes extinct, that will typically happen within just a few generations. And so because Cn is much smaller than n, in this case, most likely there will not be time for any of these type 1 individuals to enter the dormant population before the type 1 goes extinct. Now, let's suppose the type 1 does succeed in spreading to most of the active population, and then let's suppose that there are no more mutations before the winter arrives. So at the beginning of the following winter, the active population consists mostly of type 1 individuals, but the dormant population will contain both type 0 individuals and type 1 individuals, or at least the parameters can be chosen so that that will be the case. The type 0 individuals will be the individuals that have been in the dormant population since early in the previous summer, whereas the type 1 individuals will be the ones that became dormant after the mutation that caused type 1 to spread through the active population. But then early in the winter, some of these type 0 individuals are going to move from the dormant population to the active population, and some of those individuals are going to be type 0. And because type 0 is fitter during the winter, those type 0s will quickly spread to most of the active population. The time that takes is going to be of order log n, and because log n is much smaller than the length of a season, or than the length of a year, this means that the type 0 individuals are going to spread through the active population very early during the winter. So then another mutation may cause one of these type 0 individuals to become type -1, and then type -1 has the chance to spread through the active population. But then the following summer, these type 1 individuals that are remaining in the dormant population will spread to the active population and quickly take over most of the active population again. But then early in the following winter, the type -1 individuals will again enter the active population from the seed bank and spread through most of the active population again, and this process continues. So here's a picture of this process prepared by one of my co-authors, Adrian Gonzalez Casanova. So in this picture, the blue represents the summer and the red represents the winter. A blue X indicates a positive mutation that's beneficial during the summer, and a red circle indicates a negative mutation that is beneficial during the winter. And what's plotted here is a graph of the dominant type in the active population as a function of time. So at the beginning, type 0 is dominant in the active population, but then we encounter a positive mutation and type 1 becomes dominant. Then here we encounter a negative mutation, but because the negative mutation is deleterious during the winter, it most likely gets quickly lost from the population. But then this positive mutation causes type 2 to take over in the active population. So then the winter begins, and type 0 individuals enter the active population from the dormant population and quickly become dominant. And then these 3 negative mutations cause type -1, then type -2, then type -3 to become dominant. But then type 2 reemerges at the beginning of the following summer, and these 3 positive mutations then allow the dominant type to become 3, then 4, then 5. Then at the beginning of the next winter, type -2 enters the active population again and quickly spreads, and then it mutates to type -3 and type -4, and so on. And so what we see are 2 waves of adaptation moving in both directions, and we believe that eventually the fact that we have these 2 waves moving in opposite directions could eventually contribute to speciation taking place. Now, to start to state our results more precisely, we need to introduce a bit more notation. We're going to let xkn of n, xkn of m be the fraction of the active individuals in generation m that have type k, and ykn of m will be the fraction of dormant individuals in generation m that have type k. We'll let delta be the space of all doubly infinite sequences of non-negative real numbers that sum to 1. And then we're going to let boldface xn of m be the delta-valued random variable whose kth coordinate is xkn of m, and boldface y sub n of m will be the delta-valued random variable whose kth coordinate is ykn of m. So the idea is that for every value of k, we record the fraction of the active population that has type k. We put those numbers in a sequence, and we call that x sub n of m. So that records the composition of the active population in generation m. And then we do the same thing for the dormant population. Now we're going to be interested in 3 different time scales. One important time scale, of course, is the length of a year, which is u sub n. The second important time scale is the time between beneficial mutations. So let's let h of u be the survival probability for a Galton-Watson branching process whose offspring distribution is Poisson with mean 1 plus u. And then we'll let rho sub n be n times mu n times h of Sn. So in each generation, there are n individuals in the active population. They're all acquiring mutations at rate with probability mu sub n. And if there is a beneficial mutation, the probability that it succeeds in spreading through the active population is approximately h of Sn. So rho sub n represents the probability of a successful beneficial mutation in a particular generation. And therefore, the expected number of generations between these events should be approximately 1 over rho n. And that's our second important time scale. And then the third time scale is the scale on which the active population enters the seed bank. So in each generation, we have C sub n individuals moving into the seed bank, and the size of the seed bank is K sub n. So each generation, a fraction C sub n over K sub n of the dormant population gets replaced. And so the number of generations until a positive fraction of the dormant population is replaced is of the order Kn over Cn, and that becomes our third important time scale. Now for our limit theorems, we're going to scale time by a factor of rho n, so that one time unit corresponds to 1 over rho sub n generations. Now we're going to focus primarily on two parameter regimes. In regime 1, the three timescales that I mentioned on the previous slide are all comparable. So we assume that Cn over rho n times Cn over Kn times rho n converges to some positive constant alpha. This means that 1 over rho n, which is the time that we have to wait for a beneficial mutation, is the same order of magnitude as Kn over Cn, which is the time scale on which the seed bank is changing. And then we also assume that rho n times un is converging to some positive number theta. That means that 1 over rho n is the same order of magnitude as the length of a year. Now in regime 2, the three time scales are different. Kn over Cn is in between un and 1 over rho n. So this means that it takes many years before a substantial fraction of the seed bank gets replaced, but then it takes much longer still before the next beneficial mutation occurs. And as a result of that, the active and dormant populations are going to have time to reach a sort of equilibrium before the next beneficial mutation comes in. Also in regime 2, we need this additional technical condition right here. I'm not going to explain this condition in detail, but basically this condition is needed to ensure that there's enough mixing between the active and dormant populations. It turns out the condition is automatically satisfied in regime 2, but it's an extra assumption that we need. Sorry, it's automatically satisfied in regime 1, but it's an extra assumption that we need to impose in regime 2. So now we're ready to state our first limit theorem concerning the times at which new beneficial mutations spread through the active population. So recall that in regime 1, rho n times un converges to some number theta. So un is the length of the length of the year. So this means that after our time scaling by rho n, a year lasts for theta time units. Also remember, Vn over un converges to beta, so this means that rho n times Vn, which is the length of the summer after the time scaling, is going to be beta times theta. So we're going to let r of t be equal to 1 if t is between j times theta and j times theta plus beta times theta for some non-negative integer j, and otherwise r of t will be equal to 0. So basically r of t is equal to 1 in the time intervals corresponding to the summer and 0 in the time intervals corresponding to the winter. Now in regime 2, the season is changing much faster. The season is changing very fast on the time scale that we are looking at. So as n gets larger after the time change, the season is changing faster and faster. And then in the limit, if we consider any time interval of positive length, it's going to be summer a fraction beta of the time and winter a fraction 1 minus beta of the time. So in regime 2, we're just going to set R of t to always be equal to beta. Now we're going to let n plus and n minus be independent inhomogeneous Poisson processes whose intensities are given by R and 1 minus R. So in regime 1, this means that n plus has intensity 1 during the summer and 0 during the winter, and n minus has intensity 0 during the summer and 1 during the winter. In regime 2, n plus has intensity beta all of the time, and n minus has intensity 1 minus beta. Now we'll let t0 be 0, and then for positive integers k, tk will be the first time that the Poisson process n plus reaches k, and t minus k will be the first time that the Poisson process n minus reaches k. And so the times tk for positive k correspond to times when a positive mutation spreads through the active population during the summer, and the times tk for negative k correspond to the times when a negative mutation spreads through the active population during the winter. So we have the following theorem. So we fix any number a between 0 and 1, and we let skn of a be the minimum of the set of all m such that xkn of m is greater than a. So it's the first time that the fraction of type k's in the active population is at least a. And then these times skn of a, scaled by rho n, converge in distribution to these times tk. And this theorem holds in both regimes. The difference between the two regimes is reflected in the difference in the intensities of the two Poisson processes. So now that we have a limiting result for these quantities skn of a, we consider the composition of the active population. So recall that when a new type takes hold in the active population, it spreads to most of the active population very quickly. So most of the time, one type will be dominating the active population, except for short periods of time when a new beneficial mutation is spreading, and short periods of time at the beginning of each season. So during the summer, we're going to let dna of m be the largest value of k such that skn of a is less than or equal to m. So that corresponds to the largest type that has spread through the active population up to this point. And if m is in the winter, we let dna of m be the minimum of the set of all k such that skn of a is less than or equal to m. Keep in mind that's typically a negative number. So if that equals -3, that means that up to this point, the types -1, -2, and -3 have spread through the active population during the winter. And so the point is that we expect dna of m to be the dominant type in the active population in generation m. So this leads to the following limit theorem, which holds in both regimes. So we can compare xkn of m, that's the fraction of type k's in the population in generation m, to the indicator of the event that dna of m is equal to k. And the theorem says that this is a good approximation in the sense that if we consider a number of generations which is of the order 1 over rho n, then the average difference per generation between these two quantities converges in probability to 0 as n goes to infinity. So indeed, type k is dominating the active population when dna of m is equal to k, and during most of the rest of the time, type k comprises only a very small portion of the active population. The difference between these two quantities will be large only during very brief transitional periods. Now in regime 1, we can reformulate this result in terms of convergence of stochastic processes. So recall we had these Poisson processes n plus and n minus, keeping track of the times when new types spread through the active population during the summer and winter. So we will let d of t be equal to n plus of t during the times corresponding to the summer, and we'll let d of t be the negative of n minus of t during the times t that correspond to the winter. And then we'll let boldface d of t be the random sequence whose kth coordinate is the indicator that d of t is equal to k. So d of t is corresponding to the type that dominates the population at time t. And so in regime 1, we can say that these processes xn, which keep track of the evolution of the active population over time, with time scaled by rho n, converge in distribution as n goes to infinity to this process d. And here the convergence is in the sense of weak convergence of stochastic processes with respect to the Meyer-Jeng topology. Now I'll explain briefly why we're using the Meyer-Jeng topology. So let's let E be a metric space. Then a sequence of CAD-lag functions Fn converges to F in the Meyer-Jeng topology if for all positive times t and all positive numbers epsilon, the Lebesgue measure of the set of all x between 0 and t, such that the distance between Fn of x and f of x is bigger than epsilon, is tending to 0 as n tends to infinity. So consider these functions Fn, which are equal to 0 when t is less than 1 plus 1 over n, and 1 when t is bigger than 1 plus 1 over n, and the functions gn, which are 0 to the left of 1, 1 to the right of 1 plus 1 over n, and linearly increasing in between. Now both sequences of functions appear to converge to the function f infinity, which is 0 to the left of 1 and 1 to the right of 1. However, in the Skorohod J1 topology, which is what we usually use for convergence of stochastic processes, the functions fn converge to f infinity, but the functions gn do not. And the reason for this is that for functions to be close in the Skorohod J1 topology, they have to be close in the supernorm after a small deformation of time. And that can be achieved with the functions fn by making the deformation of time that moves this jump over to time 1, but the functions gn will not be close to f infinity in the supernorm for any time change, because gn is equal to 1/2 at this point, whereas f infinity is always either 0 or 1. And the scenario that we have corresponds more closely to the scenario with gn, because when a new type spreads into the population, it increases its share of the population very rapidly, but not all in one jump. Now in the Meyer-Jeng topology, the functions gn do converge to f infinity, because for functions to be close in the Meyer-Jeng topology, they only need to be close outside a set of small positive Lebesgue measure. So this topology was introduced by Meyer and Jeng in 1984. Tom Kurtz worked out some of its properties, and it was recently used in the population genetics context in this 2022 paper on symmetric coalescence. So now let's consider the composition of the dormant population, and we'll start in regime 1. So suppose in some generation, the fraction of type k's in the active population equals x, and the fraction of type k's in the dormant population equals y. So then we want to consider the fraction of type k's in the dormant population in the next generation. So there will be Cn individuals that enter the dormant population from the active population, and roughly a fraction x should be type k. The remaining Kn minus Cn dormant individuals are holdovers from the previous generation, and roughly a fraction y should be type k. And we can rewrite this fraction as y plus x minus y times Cn over Kn. So when we rescale time by rho, we see that the rate at which the fraction of type k's is changing is x minus y times Cn over Kn times rho n. And in regime 1, this fraction here converges to alpha. So this leads to the following limit theorem in regime 1. So we're going to define a delta-valued stochastic process y. Y of 0 will be the sequence whose 0th term is 1 and whose other terms are 0. Because at time 0, all of the dormant individuals have type 0. But then the process y will satisfy this differential equation here. The derivative of y with respect to t is alpha times d of t minus y of t. And recall that d of t is the distributional limit of the processes xn that describe the composition of the active population. So this differential equation is compatible with the differential equation that we derived informally here. And the theorem says that in regime 1, the processes yn, which keep track of the composition of the dormant population, under our usual time scaling, converge as n goes to infinity to the process y. And this time, because the limit process is continuous, we do get convergence with respect to Skorohod's J1 topology. Now, regime 2 is different because the time scales are not comparable. The season is changing very fast on the time scale of interest, and so we might as well just assume that it is summer a fraction beta of the time and winter a fraction 1 minus beta of the time. Also, the active and dormant populations are mixing very quickly on the time scale that we're working on. So in particular, Cn over Kn times rho n is going to infinity in regime 2. And so the differential equation on the previous slide is going to converge to its large time limit long before the next beneficial mutation occurs. So in this regime, we define the limit process in the following way. So suppose that k is positive, and suppose t is between tk and tk plus 1, then type k will be dominating the active population during the summer. And because the active and dormant populations are mixing very quickly, this means that nearly all of the individuals in the dormant population that entered the dormant population during the summer will be type k, and that should be a fraction beta of the total dormant population. If k is negative and t is between tk and tk minus 1, then type k is dominating the active population during the winter, and therefore should be a fraction 1 minus beta of the dormant population. Type 0 individuals enter the dormant population during the summer when t is less than t1, and during the winter when t is less than t sub negative 1, so we get this formula for the fraction of type 0s in the dormant population. So we can let boldface y of t be the random sequence whose kth coordinate is yk of t, and then in regime 2, these processes yn, under the usual scaling, converge in distribution to the process y. This time we once again have jumps in the limit, and so the convergence is with respect to the Meyer-Jeng topology. Now, I want to mention one technical tool that is useful in our proofs for the results for the dormant population, because I think this technique may have wider applicability for these seed bank models. So let's suppose we label the dormant individuals by the integers 1 through k sub n. And then let's let m sub I be the number of generations for which the ith dormant individual has been dormant. Then because we replace a fraction c sub n over k sub n of the dormant population in every generation, the distribution of m sub I for any particular I is exactly geometric with parameters cn over kn. So now let's let capital T be any finite subset of the positive integers, and we will let z sub t be the number of individuals the number of dormant individuals that entered the dormant population in one of the generations in t. And then using these geometric probabilities, it's not hard to calculate the expected value of zt, because we have k sub n dormant individuals, and for each of these dormant individuals, the probability that it entered the dormant population in one of the generations in t can be obtained by summing up these geometric probabilities. And so we get c sub n times the sum over generations in t of 1 minus cn over kn to the power m minus 1. Now, these events here that mi is in t, those events are not independent, because exactly the same number of individuals are entering the dormant population in every generation. However, one can use an argument based on Harris's inequality to show that those events are negatively correlated. And as a result, we get a rather powerful large deviations type bound for the probability that zt differs by very much from its expectation. And so we have very good control over the generations during which the dormant individuals entered the dormant population. And this is quite useful because suppose we know that a certain type dominated the active population during a certain set of generations. Then if we know how many of the dormant individuals entered the dormant population during those generations, we know how many individuals of that type there are likely to be in the dormant population. So I want to end by showing a few simulation results. We ran some simulations when the active and dormant populations both had size 10,000. Cn is equal to 10. The winter and summer both have a length of 250 generations. Sn is equal to 0.1, and the mutation rate is very small. So these parameters are roughly compatible with regime 1. And so what's shown here are histograms of the distribution of types in the active population at 4 different time points. So in the top left, we have the active population at the end of the 10th summer, and we see that type 5 is dominating the population. But at the end of the following winter, type -5 is dominating the population. At the end of the 40th summer, most of the active individuals have acquired around 15 positive mutations. By the end of the following winter, most of the active individuals have acquired about 15 negative mutations. And we also have corresponding results for the dormant population. So after 10 years, the dormant individuals mostly have types between -5 and 5, and there are still some dormant individuals around with types close to 0. After 20 years, there are now very few individuals left, even in the dormant population, with types near 0, and we see that these 2 distinct clusters have formed. And these 2 clusters get further away after 30 years, and further away still after 40 years. And I'll stop there. Thank you.
Thank you very much, Jason. Other questions? Yes, please, Peter.
Thanks, Jason, for this very nice talk. So in these classical papers by Ota and Kimura, they had these surprising terms for balancing selection and additional diffusion terms, and they do not appear here at all. But I think the scaling is much different, right?
Yes, I believe that's true. I mean, the scaling is set up here to be pretty simple so that you're only considering one beneficial mutation in the population at a time. We're going to hear in just a few minutes what happens when some mutations compete. Yeah, so we avoided that complication here, and so the calculation that I was referring to by Ota and Kimura is just the one basically for the duration of one selective sweep, the 2 over S log n that we're familiar with.
I have a question if there's no other question. So the dormancy is a way of avoiding the deleterious mutations in the wrong season to be wiped out by. Okay, isn't there another way of assuming that like avoiding dormancy, the dormancy ingredient, by assuming that, for example, the mutation rate are at the polynomial scaling of Dorret-Mayberry, so that they stay at a very low frequency, but still they're still there, and they can regrow when they're in the right season.
Okay, let's see. So if you're in the Dorret-Mayberry regime, okay, so certainly if you have right, so if you have the selection happening more slowly, you have weaker selection. It takes longer for the individuals to take over. And so, yes, you could get something. Okay, so what you want to do is to have the same thing with the the same kinds of mutations. Some are beneficial in the summer and deleterious in the winter, and some are the other way. But you don't want to have dormancy, but you want to have this other scaling. And I think what you're saying makes sense to me, that it should still have a good chance to work. I haven't thought about the details, of course. Yeah.
Maybe Charlene would have a sense of the answer. No? Okay.
I find it a bit strange this symmetry between a plus and minus. I mean, in particular, are there many species who become dormant in summer?
I don't know of that specifically. I don't know the answer to that. We were mostly trying not to model a particular population, but to try to establish the principle that
Yes.
You could get kind of adaptive waves going in two different directions through this mechanism. But yeah, I don't know of a particular example of.
But maybe there are other mechanisms who would give something less symmetric between the positive and negative. I mean, different from the picture, which is just here.
Yeah, I mean, you can certainly consider different variations.
Just maybe a brief comment to this. So this is a type of dormancy which would correspond to bed hatching, right? You become dormant or not irrespective of the state of the environment, right? It's not a responsive dormancy where you always go into hibernation during winter.
That's
and
right.
then you are alive. So it's, and this is a kind of an important distinction, right? I mean, so it's we call this sometimes spontaneous dormancy or bed hatching if the switch into dormancy is independent of the state of the environment, right? That is the situation here. And if you would have a responsive switch into dormancy, depending on the state of the environment, probably different things will happen now.
Yeah, that's a good point. Thanks. But yeah, I mean, we still see this effect, even though the dormancy, the effect of dormancy isn't being. I would think that in the case where the going into dormancy is responsive to the environment, if anything, this would be even easier for this to happen, right? But it happens in this model even without that.
Any other question or comment? If not, let's thank again our speaker.